Thank you. I work at the research center in Jülich, and I work at the Department of Neuroscience and Medicine. And one goal of our department is to build a three-dimensional atlas of the human brain. And my master thesis deals with one particular region of the brain, which is called the bed nucleus of the stria terminalis, and also the method of cytoarchitectonic mapping. So the bed nucleus of the stria terminalis is located in the frontal part of the brain. You can see here a left hemisphere of the brain, and the black bar marks the region where the histological sections you can see here underneath are taken from. And as this area is quite small, I enlarged the region of interest. And the bed nucleus you can see here, and also its adjacent stria terminalis. All structures you can see here are, and I will show, can be found in both hemispheres, but I only colored them in the right hemisphere. So the bed nucleus is functionally involved in stress situations, in anxiety and fear response, and also in motivation and reward systems. For the mapping, it was important for me to delineate the bed nucleus from its neighboring structures. These are, for example, the lateral ventricles, quite prominent, the nucleus caudatus, or in more caudal sections, the thalamus. The lateral border was the internal capsule. The ventral border is the anterior commissure. You can see it almost decussating. And in more rostral sections, it's the nucleus accumbens. And finally, the medial border is the fornix. And last but not least, there is the globus pallidus. It's not directly adjacent to the bed nucleus, but it still should be mentioned here as it's visible. So the method of cytoarchitectonic mapping, as I said, it was important to delineate the bed nucleus from its neighboring structures. These were based on cytoarchitectonic criteria, such as the shape, the distribution, or the size of the neurons. And based on these criteria, I could detect 4 different subdivisions. 3 were part of the lateral bed nucleus, and finally the medial bed nucleus. Here you can see, for example, the laterocentral part. You can see the fusiform and triangular shape of the neurons. The yellow laterodorsal part, it consists mainly of those cell aggregates and clustered round-shaped cells. The lateroposterior part contains a lot of stria terminalis fibers and some bigger darkly stained neurons. And finally, the medial part, which has a general very heterogeneous appearance. As you can see here, the histological sections were stained for cell bodies. It's a silver staining method. So here I prepared a short movie. After the delineation, the 3D reconstruction was done. And so you can see the actual shape and size of the central subdivision of the bed nucleus. The yellow laterodorsal part is transparent. Yeah, so far I mapped 7 brains, and in total it will be 10 brains. And after the reconstruction of all the 10 brains, the reconstructed structure will be warped to a reference-based template brain, and we will build so-called probability maps. So that's it. I delineated the bed nucleus on high-resolution images. I detected 4 subdivisions of the bed nucleus. A surface-based reconstruction was performed, and in the future we will transfer these to a reference space and build probability maps to gain one additional step of the human brain atlas. Thank you.